have heard of the word transcription before. Transcription comes from the root word transcribe, which means to put thoughts, speech, or data into written or printed form. This might have been done by you in class or maybe at home. The usual example of transcription is when you're asked to listen to an audio and then you would be writing down what you hear. It does not only happen in our daily lives, but it also happens within our DNA. But in the biological sense, transcription is part of the central dogma of molecular biology. This is the one that comes after DNA, or I mean DNA replication, because the source of an RNA or the template of RNA can only come from the DNA. That's right, I know, I heard you. Telepathy. Anyway, by definition, transcription is a process that involves copying a gene's DNA sequence to make an RNA molecule. We have to take note of the phrase DNA sequence. Why? Because transcription does not require the whole length of DNA to be opened up and copied in order to create or produce one protein only. Much of the things that we know are based on studies done on Escherichia coli. We have introduced this bacteria in our class a long time ago. They are a scientist's favorite per se. And the one in the next bullet, it says here, occurs in individual genes. So only those genes that code for specific protein will be opened up when transcription is occurring. Why? Because not all of the genes that we have are not needed at that moment. Okay, so let's say by this time, I need a protein that will increase my amylase in my digestive system. So the only part of the DNA near my digestive system or my mouth that would open up would be the area that can produce or that codes for amylase. Okay, in the image in here, you could see that this area is highlighted or colored with 1. It means that this is for a specific protein. This link in here in yellow would be for another protein, and another one will be for a third protein, a hypothetical one. So we could also say that the length of each gene that would be used for transcription will not be the same. Some gene might be longer, some gene might be shorter. And in relevance to that, the number of RNA molecules for each gene may vary. Their needs or the types of proteins involved for each gene are different. So there is no specific number in average. But for that one particular gene, there is only a set number of protein that should be produced. If there are more, or if there are less, or if they missed a DNA sequence or RNA sequence, that could mean problem for your cell. Next one. Nucleus to cytoplasm. Transcription will occur in the nucleus because the DNA is inside the nucleus, and the product mRNA will be traveling to the cytoplasm. Okay? So, next one. Focusing on this box in here, why do we need RNA? Okay? We need RNA not just because our body wants to, or whoever created us think that it's just a good thing to have an extra step, just, in case something happens. But if you think of it, if the DNA is the master copy of everything in our body, why not just have the DNA do the job? Having an RNA or producing RNA would take time, right? If you take it that way. But there is a reason why we have to use RNA. Number one, ribosomes cannot enter the nucleus. The nucleus is overprotective of itself. It does not let anything go in or out, just like a cell wall of a plant, which is super permeable to anything. Okay? Number two, DNA cannot be decoded directly. This one is related to number 4. This is because ribosomes can only read mRNA. Wherein in RNA, as we all know, it's not ATCG, right? Rather, it will be CG, but the A will be partnered with letter U for uracil. Next, number 3. DNA cannot leave nucleus because if the DNA can even go out, there will be a lot of factors or substances present that can damage and cause mutation to the DNA. And on another note, DNA is too big and too long to even be able to go out of the nucleus. Moving on to this box. Okay, so looking at this box, you could see again the word sense and antisense. You were asked to familiarize yourself with this in the previous form. And the sense strand, just a recall, runs from 5' to 3', and antisense will be running from 3' to 5'. The coding strand is this, and the template strand is this one. This is a sample of how will the mRNA look like based from the template itself. So for the template, it's written here TACCTT. Let's cut it here. As you could see, through the rules of nitrogenous base pairing,
T will become A, A will become letter U. It will not be a T anymore because letter T is for DNA. It is always replaced by uracil with the symbol letter U for RNA. C would be G, C and G. They're still the same, just like in DNA. T and then it becomes A, T, A. A again becomes letter U. A will be U. Letter G will become C. A will be U. G to C, C to G, G to C, A to letter U, and G to letter C.
we will be having what do you call this one? An exercise at the end of the form for this one. Moving now to the most important part of transcription, which is the process. The process. So in the process, it's not as complicated or it doesn't have as much enzyme compared to DNA replication. For transcription, we only have to remember one special enzyme, and that is called as RNA polymerase. We will describe that enzyme later on. For the first one, a section of DNA coding for a protein, which is the gene, would unwind due to breaking of hydrogen bond between the nitrogenous bases. So in here, we have the DNA itself before it is unwounded or before it is unzipped. And as you could see in here, you will notice a red line, and this one is a set of blue lines. These two lines are important because they would tell the RNA polymerase, where to start and where to end their process. They are the starting and they are the ending. But specifically, the red one in here is called as the...
This one is called the promoter. And the blue one in here is known as the terminator. Just like Arnold Schwarzenegger movie.
So, from the name itself, promoter, it promotes the RNA polymerase where to start the transcription. It is made up of nucleotides that do not bear any information. And the terminator, on the other hand, people, from the name itself, terminate, meaning this one will signal the end of the transcription. Once the RNA polymerase reaches this terminator, it will be a signal for the mRNA that it is already complete in that particular process. Please again note the coding or the sense strand is the template of the mRNA. Next one. Next step. DNA polymerase attaches to the promoter region and moves from 5' to 3' as it adds new RNA nucleotides. Please bear in mind that the RNA nucleotides are A paired with U, C will be paired with G. And there will be no hydrogen bonds between the mRNA that will be produced and not the polymerase, but rather the template strand.
Template or coding or sense strand. They are all one and the same.
So this process is shown in here. The coding strand is highlighted in orange color, and the RNA polymerase is shown in pink circle, and the arrow accompanying it will show the direction that it would be moving. As it moves along this length of the coding or the sense strand, the RNA polymerase will keep on adding nucleotides of the RNA. Again, as it reaches the end or the terminator region, that is the only time wherein this mRNA would be detaching and moving on to the next part of the process, which is letter C. RNA polymerase forms phosphodiester bonds to make sure that the backbone of the mRNA molecule are attached together. Lastly, mRNA detaches once RNA polymerase reaches the terminator. Again, this one refers to the blue region that we saw earlier. This one. It then leaves the nucleus via the nuclear pores in the nuclear membrane. It enters the cytoplasm for translation at the ribosome, which can only be found in the cytoplasm. This image in here will show you a diagrammatic way of how it moves. Next, lastly, DNA double helix will be reforming inside the nucleus. So, the mRNA that was formed after transcription will now be floating around, looking, technically not really looking, but freely floating, wanting to meet a ribosome so that the protein can start processing now. Okay, so for the next part or the last part of protein synthesis, please go to the next video after this one. Bye everyone!